turn on your time machine in your brains and going back like 3 years ago and imagining what was happening in January 2020. If you still can recall what was in the news at the time, how people were perceiving the different things and so on. And you might remember that in January 2020, people were not paying attention much to the news in China about a new epidemic, even though it was already in the news. Some people thought it might be the next big thing, but most of people did not pay attention. And when you look back at the events, you could see that when the new virus appeared, when it emerged, the first reference genome of this virus was published very early on, already in January 2020, much before the virus spread in Europe and in the US. And in some sense, it's a big event because today in the era of genomics, once we publish reference genomes for the first time, once we sequence a new organism for the first time, there are many things we can do with it. So immediately, there were people who were working in the vaccine industry or vaccine design technologies for vaccines. And immediately when this genome was published for the first time, even though the virus didn't make it to Europe and the US yet, the immediate reaction when they see the epidemic starting and wondering that this might end up in something bigger was, we know the genome, let's make a vaccine. Because nowadays with the genome you can design vaccines very well using the new technologies which we have for vaccine design, especially the mRNA vaccines. So that was the first reaction. At the same time, as there are many bioinformaticians interested in public health, there are many bioinformaticians who are wondering, okay, so we have this genome, it's uploaded to GenBank. Has this genome been ever seen before? Can we scan, can we look into metagenomic data and verify if, for instance, the virus emerged, let's say 2 months before people observed it for the first time at the Wuhan market, for instance? And we're asking this type of questions. And for people who work in bioinformatics, this seems like a very natural question. Actually, the answer was not that simple because it turned out that it's extremely difficult to search the genome. Essentially, all the labs immediately started to use MeSH and tools like that for matching against, let's say, standard reference databases. But very quickly ran out of options, found some matches with some of the coronaviruses that are in public repositories. But looking exhaustively through whole SRA, including metagenomic data, was actually very difficult. So this example bioinformatician, which represents the whole community, would say, let's scan all the data, because we don't have any tool that would be able to search in all the metagenomic data yet. Now, let's move 7 months later, in the mid-August 2020. The first guy who represents people who work on vaccine design at this point could say, my vaccine already passed phase 1-2 trials, and ask the bioinformatician, what about you? And it's the time when this was published in Nature. In fact, the paper was submitted even before. And the bioinformatician would say, just finished my search of the genome in SRA. It was the time when the Ceratus project was preprinted, or the results of the Ceratus project were preprinted for the first time. Eventually, this work was also published in Nature. But it just shows the huge delay. It was essentially faster to design a new vaccine than to verify if the genome has ever appeared in metagenomic data before. And if we also imagine a politician or an expert in public health who decides about public health policies, this person could say, too late, it's no longer actionable. Because you need the answers like immediately. Like the answer whether this virus, well, the origin of the virus were especially relevant at the beginning of the pandemic when people wanted to understand the behavior of the virus, where it emerged, and so on. So how does currently search look like? What is the state of the art? Like when you talk to biologists, they still use the old school way to search sequencing data. They use BLAST. It's actually a very good tool that was developed in the early days of, I would say, DNA sequencing, or at the beginning of 90s, where still the amounts of data were very small. In fact, we speak about it, or we always say a tool. It's a huge collection of different tools. It does allow many different types of searches. It's incredibly flexible. The way how people typically use BLAST and there are many different ways, which is always important to keep in mind, is that we have new sequence of interest. For instance, we have a new virus, we have a new bacterium, some gene, whatever. And our typical question is to ask, what is the most similar thing that has been seen before, for which we already know the properties? And BLAST can answer this extremely well. And there are actually a lot of methodological development that were done. So it can first give you results in form like a little bit like Google as like a list of the top hits. And for all the top hits, because we typically are not interested in, let's say, 100,000 different results. In most of the cases, we are interested in just the most similar hits. And we want to manually pick up the most interesting genomes in the reference database or genes, and then carefully look at them. And then we are interested in the specific alignments, and we are also interested in various things such as e-scores and so on to understand if possibly this alignment emerged randomly or not. The big difficulty is that even though the BLAST works extremely well, people are very much used to use it, especially biologists. When you talk to biologists, they are using BLAST essentially every day. It's their main tool. The difficulty is that the database is small and not even remotely scalable. The whole framework is not even remotely scalable to modern data. On the other hand, it really acts as kind of like a Swiss knife. So we have like a small database, but then we can do many different things with many different, let's say, tools. By different tools, I mean different search algorithms for proteins, for nucleotides, with different levels of divergence. And since then, there were many, of course, the whole BLAST framework was improved many times. There are progressively new papers about moving to the cloud, about better algorithms for, let's say, matching and so on. So the original BLAST that was published many years ago, several decades ago, in the 90s, is very different from the BLAST we are using nowadays, even though the fundamental ideas still the same. So since there were like huge, I would say, revolution in data search, and many approaches were designed to increase the search capacity. Actually, the way to think about it is essentially going from the Swiss knife, which we have at the beginning, the BLAST, that's applicable for many different types of questions and can be applied with different levels of divergence, to tools that have, let's say, higher capacity but are much more specialized and are typically designed for like one type of queries. And just to mention some of them, and there are many, there are amazing keyword-based tools such as Bixby, COPS, Metagraph, and so on. That actually simplified the search, the original question of the alignment, to much simpler problem of matching keywords. Many others. And with this type of tool, it became finally possible to search in much bigger databases, not just in the curated databases of BLAST. We have also full alignment-based approaches. And on this front, we can highlight, for instance, Minimap2, but many other tools. This is a super active area of development. But none of these tools actually replaces the original BLAST. There's another problem, and this problem is that even though we were able to increase the search capacity a lot, still these were some kind of like one-time improvement. All these tools are struggling exactly with the same issue as the original BLAST tool was, and did not manage to somehow like overcome one thing that we will mention on the next slide, essentially going from like this one, let's say, specialized tool, somehow applicable, to much more general tool, where what I want to highlight with this figure, which like illustrates that some tool to which we would like to get, is that it's some kind of like a universal engine that we can combine with many different tools with many different bits for different types of searches, where the engine itself can work on very high scale. And at the same time, we will be able to adjust the tools for specific types of biological questions and specific types of data. And the fundamental problem that's behind all of these tools is that the number of genomes grows vastly faster than computer capabilities. And this is a very well-known fact. So just to illustrate this, and now please look at the trends in this graph. I will explain what's there. So on this graph, on the x-axis, you have a year. On the y-axis, you have some unit. Units are not important at this point. We are interested just in the trends. So on the top, we have the number of bacteria in the NCBI assembly database, and it's growing exponentially. The doubling time is approximately 1.3 years. The orange curve is the iPhone storage. I chose iPhone as like typical representative of some specific technology for which we have data for some time, and where we can observe certain trends. How the memory is increasing, how the computational capacity is increasing, and so on. And the third curve is the BLAST-NT database, how it was changing in course of time. And what you can see in this plot is that the iPhone storage is essentially aligned in terms of growth with the BLAST-NT database. And both of these curves go much slower, and it's logarithmic scale, don't forget about it. Both of them go much slower than the number of bacteria that we have on NCBI. And this has a profound consequence. This essentially means that the proportion of data, published data, that are actually searchable is exponentially decreasing over time. This is a plot which demonstrates essentially the size of the BLAST-NT database over NCBI bacteria. It's extremely difficult actually to find, for instance, the proportion of bacteria in BLAST from technical reasons, but still we can compare the trends and we can see in long term how these two quantities are changing in course of time. And for obtaining this type of plot, actually, it was extremely challenging. We really had to do some kind of archaeology, look into old papers, look at how the BLAST database was changing in course of time. Because if you have ever downloaded the NT database, you will see that it's changing every several days. And there's actually almost zero reproducibility. You have no way how to return, how to get back to the old versions of the BLAST-NT database. So you have to look into literature, you have to look into different sources, and some kind of like reverse engineer a little bit the original size from public data. We can also look on the logarithmic version of this plot, and then you see very clearly the exponential decrease, because then this curve becomes almost linear. With one interesting exception, which is the change of the curve around 2020. And in this case, we still don't know if it's just an artifact in the data, because it might be some data get to the archives a little bit later. So sometimes the ends of these curves might not be completely accurate. But also it might be some technological change, where BLAST, for instance, by better use of polarization or something, slightly improved for some time. But the long-term trend is very clear, it's exponential decrease of searchability. So now the question is, how to beat this exponential decrease? Because for this, we need some fundamental innovation. And the first thing, if you work with, let's say, big data, and we start speaking about complexities and how to compress them and so on, the key thing is we have to move towards the concept of entropy. This becomes a little bit tricky, because there are several different entropies that are not compatible. So when in the context of bioinformatics, when people say entropy, you should always ask which specific entropy they mean. Because typically people speak about entropy in the way, in the definition, let's say in the information theoretic style, think of Shannon and channels and sending messages over channels. And in fact, this type of entropy is not extremely useful. From one reason, it does not capture the repetitive nature of data. If you have a string S, and then if you double the string, if you concatenate it twice, this would require twice as much space, even though the information there is in fact exactly the same because of the repetition. So this concept is in fact not extremely useful. On the other hand, there's another entropy that's used when studying algorithms for search. And it's the magic entropy, which refers, and I won't go to the full definitions, but essentially it refers to the smallest number of balls that can cover your genomes in some high-dimensional space. So the way how to think about it is to think about genomes as points. Imagine a super high-dimensional space, and distance between the points can be, for instance, at a distance. Of course, let's say, going to, let's say, this mathematical formalization is a little bit tricky, because of course at a distance is not the best possible distance, But for simplicity, we can keep it just for the illustration of the concepts. And actually, the fundamental discoveries about how to deal with these exponentially growing data, growing faster than computers, were already done approximately 10 years ago in the group of Bonnie Berger at MIT. And they had a whole series of papers where they came up with the concept of compressive genomics and so-called compressed entropy scaling algorithms for search in genomic data. The first key idea is that if we want to keep up, we cannot search in the uncompressed data. We somehow have to move to compressed representations and search directly in the compressed representation. And this was published in Nature Biotech in 2012 with example tools and many things around that. Another paper from the same group that then came up with very good mathematical formalization was published in Cell Systems 4 years later. And in this paper, among many other things, there are a lot of interesting information, ideas, and so on, especially in the supplementary. They came up with the observation or the idea that genomic data, future low magic entropy and fractal dimension, which are some of quantities for measuring properties of magic spaces. And these, if we know that these two values are low somehow, which we know for genomic data, or they have shown it on some examples, this guarantees the existence of efficient search algorithms. So thanks to this work, we know that the efficient algorithms exist. Now the question is how to somehow turn them into practice, because there's always a very long path from the original ideas, let's say some kind of conceptual / mathematical ideas, to something that can be used in practice. And what's actually the practical challenge? The big practical challenge is just how to identify redundancies. It seems simple, it's actually extremely complicated. Think of very big collections, let's say millions of genomes. If you are asked to identify redundancies between these genomes, it's a very difficult, challenging task. So it's a problem that has been studied for extremely long time. There are many reviews that summarize all the advancements in the domain of genome compression. Here are several pointers to them. In general, there are several classes of compressors that are used and tested, and some of them are just, let's say, used for research purposes, just for seeing how far we can go, but are not practical at all. Some of them are even super practical. In general, we have one class of compressors that's called dictionary compressors. Most of these tools are based, or methods are based on implicit, but not all of them. Typically, Burroughs-Wheeler transform-based methods are classified into this group as well. And the fundamental idea behind is storing repetitive phrases within a dictionary. And many of the tools that you use probably on a daily basis, like gzip, bz2, lzam, and so on, they belong to exactly this category. There are also specialized compressors that were designed specifically for genome data, such as MBGC, multiple bacterial genome compressor, which is specifically tailored for the structure of bacterial data, especially for reverse complements. Reverse complements is something that can be supported in general compressors, and it can actually make a difference. Another big class are statistical compressors, and they are currently studied also thanks to all the developments on the side of neural networks. These are extremely efficient in the resulting compression ratios, but very slow, so not very applicable in practice. But with this type of compressors, you typically can get better results than the dictionary compressors. And then I would like to also mention one category which is a little bit different than the others. That's tools for camera matching, camera indexing, and so on, because this is a whole subfield of bioinformatics with a lot of development and tools such as recently introduced Temisto, or for example, for instance, Fulgor. You will see why I'm including these tools as well. Anyway, the general issue is that the compression is generally extremely difficult to scale up. It's typically possible to apply the compression on, let's say, small quantities of data or low diversity data, but it's very difficult to scale it up to whole genome collections. The main reason is that genomes, again, are growing too fast. So even though in principle we can compress them, we don't have sufficient computational power. And as a consequence, what people use still in practice is gzip, which is completely unable to go below the entropy bound, like 2 bits per base pair approximately. So a tool that essentially does not compress or compresses only to the, let's say, converting nucleotides to the 2 bits per base pair encoding. And microbes are especially hard from the reason that it's the most ancient, abundant, and diverse form of life, which also undergoes a lot of horizontal gene transfer. So microbes are very complicated, actually, when you look at the geometry. It's pretty complicated, but at the same time, as we will see, simple in certain aspects. So why is it difficult to detect redundancies for compressed data? I've prepared just one simple example to demonstrate this, but this phenomenon is general. It does not concern just K-mer matrices. So if you imagine K-mer matrices where you have, sometimes it's called annotation matrices, where you have genomes in the columns, you have K-mers in the rows, then you have presence absence, which is a typical notion that's widely used in bioinformatics nowadays in K-mer-based methods. You would actually see that for microbial data, these matrices become extremely huge, actually in both dimensions at the same time. And this is the fundamental mathematical challenge behind. Because at the same time, we are increasing the size of the genome collection, and at the same time, we are adding more things. We are increasing the diversity of the genome collection. One example from one, I would say almost now canonical data sets that are used for all the benchmarks nowadays. If you look at how this matrix looks like, it's essentially after when you put like bit compression. When you use bit compression, you put 8 bits into 1 byte. Just storing such a matrix would require like 3.6 petabytes. So the matrices are really big. And just to highlight that this phenomenon is not just about the K-mer matrices, that this is something much more general. You can think of the diversity of genome collection here, like as the set of all the K-mers. This concept as kind of like analogy to the dictionary and LZ-based methods. So essentially, if you are increasing the diversity of genome collections, it essentially means that to compress well, you would have to proportionally increase the sizes of the compression dictionaries, which would go beyond memory capacities of most computers. So what are possible strategies to achieve compressibility? How to deal with that? These are the strategies you can see in essentially all the modern all the recent bioinformatics papers about this phenomenon. So one of them is limiting the diversity. So essentially decreasing the number of rows. Typically, then your strategy is focus just on one species. Take many genomes of the same species, but you essentially know that the total diversity won't be huge. So it will become very well compressible. You make one dimension small. Or you can make the other dimension small. You can say, okay, I allow a lot of diversity, but I will decrease the number of columns. I will decrease the number of data sets. And for instance, we have done this in our paper about de Bruijn graphs and simplitics, where we show that just with BWA, we can very easily index in very rich species span genomes. We index like 3,000 species span genomes. A very similar thing was done recently in the Themisto paper, where they also went from the original data sets to species span genomes, and then everything becomes simple, because again, one of the dimensions is small. Another strategical choice might be to parallelize over many computers. It's not sustainable, and you need to be at some university with huge cluster. But this is not a solution that would work for a long time. And this is strategy that we use to use population genomics, because actually the structure of the data is very well predictable. It's not random. It's not something unknown. We know it actually quite well. And the key insight is that the genome data geometry is easily predictable from the evolutionary history. So now, if you imagine that you have a huge set of genomes you want to compress or you want to search, if you imagine that you put them onto one phylogeny and now put aside the discussion about whether there can be a single phylogeny and questions like that, just now imagine that you can for simplicity. So if you look at the evolutionary structure, you realize that it isn't almost perfectly explains the distribution of all the redundancies, or most of the redundancies among your sequence genomes in your database. Essentially, two genomes, the more closer they are within the phylogeny, it means that they are more mutually similar and they are more mutually compressible. More redundancies they have in common. It's a consequence of how evolutionary processes work. Similar ideas already appeared in the literature and were used in various domains, especially in metagenomics. The way how Kraken works and White works is based on very similar ideas. That similar genomes get concentrated in the same, let's say, taxonomic units, and then you can use very aggressive compression techniques. In the case of Kraken, it's ROC compression, and you can represent actually a huge number of genomes in a relatively reasonably small space. So we get now to the key slide. What's the phylogenetic compression of data structures? So in general, by this we mean reversible reordering of input data according to the topology of their phylogeny. So phylogenetic compression in the simplest form essentially puts genomes into the order of their similarity and makes them easily compressible, indexable using existing approaches such as wall-level tools. So it's not complicated. In principle, you just take all of your genomes you have, you look at the phylogeny where they are positioned, let's say, on the evolutionary trees, and you sort it left to right according to the phylogeny. Extremely simple as a concept. And this works for assemblies, jabbering graphs, Bloom filters, LSH indexes, for many different things, most of the representations we have. As you can see in this, it shares, the fundamental idea shares many similarities, for instance, with the Boros-Wheeler transform, which is also based just on reordering the input data, so that then when you apply additional compression methods that do the actual compression, they will work better. It's a very similar principle, just works completely differently. But you can also think about some kind of like phylogenetic transforms to boost compressibility. So just like my example, like if you wanted to do this in practice, how we would proceed before we look at how to do it for big data. We would start by inferring a phylogeny using some like method. Even mesh tree, even it's not perfect, we know from our experiments that it provides sufficiently good results in the estimation for the compression purposes. Then you would order the genomes according to the phylogeny left to right, which is extremely simple. You just essentially print the leaves of the new tree. And you would then compress it using an optimized low-level compressor, such as XZ or MBGC. This point can be a little bit tricky. You need a good compressor which has sufficiently big windows, big dictionaries, and so on. You could not use, for instance, gzip, because the window of gzip is too small. So let's go bigger now. So how to make this scalable to really big collections, such as EBI, comprehensive genome collections, which are some of the biggest bacterial collections currently available. So we are actually focusing on two collections that were published in the recent years. One is the collection from the Big Z paper, the so-called Big Z database, which consists of 448,000 microbial jabbering graphs that were computed directly from SRA, from sequencing reads, and then put into same data structure for search, so-called Big Z index, BitSlides genome index. These data, as they are published online, they have after all the error correction or all the cleaning, removing k-meres likely corresponding to errors and many other things. These jabbering graphs that you download occupy approximately 16 terabytes of data, or almost 17 in the Max Cortex format. Another collection we were very highly interested in was the 661K collection. For the reason, again, it's some collection obtained in a consistent manner. Like you download essentially all the data from SRA, all data of bacterial isolates. This is important at this stage. We don't work with metagenomes, just with bacterial isolates. Even for this, everything is difficult to scale up properly. Essentially all bacterial isolates on SRA until 2019 were downloaded and assembled in a consistent way using exactly the same pipeline. And this collection occupies, when you look in the supplementary, 800 gigabytes after GZIP compression. So how to scale up phylogenetic compression to this? We can use a key insight, we understand the structure of the data, because in population genomics, these things have been studied for a long time. And we have very good understanding about various distances between different genomes. So the most important thing is that genomes in microbial repositories form clusters corresponding to species. Whether species form clusters is unclear. It's an open biological question. Probably the answer is no. But genome in public repositories have such clusters, even though they are sometimes imperfect and two different species can be within the same cluster and so on. For computational purposes, this does not matter. We have the clusters there. That's important for us. Also, microbial species form distinct lineages. So in theory, you can even zoom into microbial species, and you can obtain clusters on lower level. And this is very important from the computational point of view. Genomes can be assigned to their respective clusters using metagenomic classification. In principle, we might be able to apply even purely classification-based approaches, purely, let's say, computational clustering approaches. There are good reasons why we want to do metagenomic classification, because if we want to use the data for essentially whatever, we need the information about contamination. Otherwise, search will not work very well later. So we can essentially take some database such as RefSeq and use it as kind of like a prior on the structure of future data. So how it works? You essentially are in a very high dimensional space. Now we plotted just for illustration, but it's super high dimensional. We have some database such as RefSeq, which are the color points here. And using metagenomic classification, for each data set, we can identify the species essentially as nearest neighbors. And effectively what we are doing from the mathematical point of view is some kind of Voronoi tiling of the genome space. From the viewpoint of computation, the important thing is, if you now imagine you have infinite number of nodes, you can do it at all one time. It's perfectly parallelizable. You can do it completely independently, and you will get these clusters. So if you have a huge cluster, this is extremely fast. And so one consequence is that it's applicable to data of arbitrary size, under the assumption that you have sufficiently many nodes. So we don't have to worry if you have big clusters. You don't have to worry that at some point in the future our algorithm will stop to work. If you return back to the notion of color matrices, what this essentially means is some kind of factorization of the color matrix. So from an original color matrix that was huge and essentially impossible to manage for big databases, we are now getting to a set of small color matrices, which are now very well manageable and very well compressible using the available approaches. So we are almost there, but we still have some difficulties. Some clusters will be too big, some will be too small. It's very natural. For instance, if you have Salmonella, you will have too many genomes there. So it's not very comfortable, or it's not very, our goal eventually is to be able to run all the analyses kind of in parallel. And we would like to establish, for instance, upper bounds on various, let's say, times of search and so on. So it's not very comfortable to have clusters of uneven sizes. So we do some kind of additional rebatching. This is just an example of a heuristic that we used for this 1K collection. You start at the beginning by the species clusters, as they are identified by metagenomic classification. And then you are actually looking at the size of the cluster, if it has more than 100 genomes or less, and so on. And based on that, on one hand, you are decreasing the size of the big clusters. On the other hand, you are merging the super small clusters into something what we call dustbin, which is a term which is sometimes used in population genomics for, let's say, genomes that are kind of different than anything else and are kind of flying like a dust. So we borrowed the term from population genomics, I would say, practitioners. So in the end, from the original, let's say, 2,600 species clusters, you will end up here with 300 batches. It's a heuristical approach in this case, but there's deep math behind, because in some sense, it's designed in a way that it optimizes at the same time let's say, the maximum size of the data set, at most 4,000. And at the same time, maximum number of k-mers, because if we have some guarantees behind. So it's not like completely random. We also might be able to study this from the viewpoint of, let's say, the delta measurers, string attractors, and so on. And we could develop like a whole mathematical formalization of this. And so now we have the batches. And now it becomes simple. So for every single batch, we can run in parallel, completely in parallel. We can perfectly like split it across a cluster. The following procedure, what we have actually already seen. So we are starting with like limited number of genomes. We infer phylogeny, and then we just sort it left to right according to the phylogeny, or we can also like kind of like propagate the signal of the genomes bottom up. Both of these approaches are actually quite similar and yield very similar results. It's not a big difference. The key thing is that we are using the phylogeny for reordering the data in a lossless way. So, summary of the method, just when we put everything together. So, phylogenetic compression has 4 ingredients. First is clustering into phylogenetically related groups, which we can do using metagenetic classification, for instance. But we could use also other approaches. There are now very interesting works about how to use GPUs, for instance, for clustering of genomes, even without like having a reference database and so on. Second step is inferring, or second ingredient, a compressive phylogeny. So already within a given batch, for which we have guarantees on the size and diversity, we infer a compressive phylogeny. And then we use this phylogeny for reversible data reordering. And at the end, we apply a calibrated low-level compressor or indexer. So then we use some of the tools that were published earlier, and can deal with the super big data. But if they get reasonably small data, they work very well. And this approach is extremely general. It can be instantiated to very different protocols for individual data types. So we played with some of them, but it can be used for many different things. So just some examples of phylogenetic what we call protocols. So for assemblies within a given batch, again, we infer phylogeny, sort it left to right, and compress it, for instance, using XZ, using the Lamp-Zip Markov chain algorithm. For the Brink graphs, we use slightly different approach. We again infer phylogeny, look at the k-mer sets, and we propagate the k-mers bottom up in the phylogenetic tree. It's actually very comfortable from certain reasons. We have been originally using this for the purpose of metagenomic classification and compressing indexes for metagenomic classification. And after we propagate the data bottom up, we compute so-called simplitics. And again, we compress them just with XZ. So very simple approach. And finally, we can use exactly the same approach for k-mer indexes. Just in this case, we have, let's say, some kind of like array at the beginning. We have the phylogeny, and we just reorder the columns according to the phylogeny. And again, we can compress it, for instance, using XZ, but possibly with many other methods too. So just one example, because this is easy to visualize. What happens when you have boom filters? If you do this, you end up with some structure like this. This is example for Neisseria gonorrhea. So you get extremely regular structure within your data, and as you can guess, this is extremely well compressible. You can even like look at these arrays or the signal you are seeing there from the viewpoint of pangenomics. So every genome, every column is one genome, Bloom filter column genome. Every line is bit, and you can even identify bits that correspond to core genomes. You can identify bits that correspond to accessory genomes. You even are able to visualize contamination and various different phenomena. It's actually beautiful. Like you can play with it a lot. By the way, this is just a side note. For some time, we were playing with algorithms for compressing images, really like PNG and so on. And we were testing them. They actually did not perform as well as XZ, but we spent a lot of time. If you ever want to test these algorithms, I recommend using Adobe Illustrator. That's probably the only efficient implementation of the compression. All of the other tools were failing because our images were just too big. Sorry, Photoshop. Okay, so now let's apply all of these ideas, all of these methods to the collections that we mentioned previously. And I will go directly to the results. We have done tons of calibration. There has been actually a lot of work about like how much of the signal is explained by phylogenies and so on. But I don't talk about this because it's actually quite technical. So when we apply this to the De Bruijn graphs from the Big Z collection, we actually managed to losslessly compress the original 16.7 terabytes of data to 52.3 gigabytes in the final archives. As for the 61K collection, this was originally the starting point was much lower because it was assemblies, which are already kind of like clean, easy to represent and so on. So even though the collections are a little bit similar, essentially you can think of 61K as a superset of the Big Z data, even though it's not completely true. The original goal of Big Z was also to include viruses. But conceptually you can think about like a bigger data set than Big Z data. So at the beginning, we were starting with like 0.8 terabytes, and with a specialized compressor as the low-level tool, MBGC, we were able to get it down to approximately 21 gigabytes by exactly the method that I described. And we are also playing, and this is just like kind of like a fun fact. I can also show. This is maybe one thing that I would like to highlight. Like whenever you speak about compression of genomes, it's always important to provide not just one characteristic, but several different characteristics. One of the characteristics is how much space you need per genome, and then how much space you need per distinct inverse. If you provide just one of these, you don't say actually much about like what's happening, because it can be the nature of the data can be completely different. And there are trade-offs. When we visualize both of these characteristics, there's some kind of like a trade-off. So either you have a very good one or you have a very good two if you are using a good compressor. So for 61K, we achieved we needed approximately 31 kilobytes per genome. And it corresponds to something like 0.06 bits on bytes per base pair, and approximately 3.7 bits per distinct inner. So these are the compression results. And we were also playing with like also some like fun ideas, and unfortunately, eventually we didn't manage to make it work. I don't know if there was issue with funding that we could not buy a pigeon, or there was something like that. But we actually calculated that like sending, for instance, like all bacteria from New York to Boston is much faster with a pigeon than over the internet. So maybe at some point in the future you will see some results in this direction. Michael actually loves ideas like this and coming up with like original technologies and so on. So we had a lot of discussions about the feasibility of this, because like if you are able to compress the bacteria a lot, you can put them just on like an SD drive. And in general, like sending data over the internet is slow, and if it's the right distance with pigeons, it's really much better. It's faster. So, and then like, just going back, natural question is, can we search in these data? And again, we can use phylogenetic compression for designing various, let's say, search protocols. So we can even do alignments with this. Can be instantiated to different search protocols with different methods and so on, just as a proof of concept to show feasibility. We focus on one specific protocol and its implementation, and where we focus on the situation where your queries are in batches, like one FASTQ file, for instance. We stream over the whole database. We decompress in memory, and we first do k-mer matching and then alignment just to the best candidates identified during by k-mer matching. But many different protocols can be imaginable. So how we actually implement it? It's implemented in a tool that's called MobSearch. By the way, if you go online to this repository, you will also find links to the other repositories related to this project, also to the repository for compression, for the actual compression, and so on. And we are still working on the software. So, as I mentioned, like for filtering, we use COPS. So we use, as we shown previously, like we can compress COPS, the boom filters. So we do exactly this. We have compressed index for the 61K collection. We decompress it in memory. We rapidly compare our queries, again, individual COPS indexes. And then we kind of like aggregate the results. And for each query, we identify the best possible candidates in the database. And for these, we do full alignment. And again, we iterate through everything, we decompress everything. Intuition of many people is that decompression is slow. No, decompression is extremely fast. That's not the bottleneck. So for us, for this type of approaches, we are perfectly fine with doing decompression. In long term, at some point, we would like to be able to search directly in like the compressed data. By the way, it would be a different game if we were interested in real-time search, and we would have to do very different, let's say, technological choices and parameters and so on. This was specifically designed for batch queries. As for database, as I mentioned, we use 661K again, but we use slightly smaller version where we excluded those genomes that did not pass quality controls. And there are good reasons for that. If you include genomes that are contaminated, it happens that these genomes act as some kind of like attractors, and you get like false positive matches. So it's important, if you want to do like good search, it's important to have like database of reasonably good quality. And this is actually something that gets very often underestimated by computer science people who are just interested in the search, like as a computer science problem. Once you move like more to the biological side, you realize that the quality control is extremely important and must be somehow like felt through at the scale of the whole project, not just at the end, for instance. And the required resources like are actually quite modest, like for disk, for everything, for like the COPS index, for the minimap, for the alignments, sorry, for the reference genomes, we need only 100 gigabytes of data on your disk, which is not that bad. So it means that for like this type of search for the alignment, you need approximately 0.3 bits per base pair. 23 bits per distinct camera, again, to provide the same type of statistics as previously. And in terms of memory, you need 14 gigabytes. So it's like reasonable for modern laptops, like you can use it on most of modern computers. They can be like too like low memory, but as long as they have like sufficient memory, it works essentially on any laptop. We did the evaluation, we did like various experiments, but I will highlight just two. The evaluation we did on an iMac with 8 cores, I think it's with like hyper-threading, so in reality it's like actually only 4 physical cores, but it looks like 8 cores. And an SSD, the size of the disk sometimes can have impact, so that's why I'm mentioning it. And as probably the most convincing experiment, we used the EBI plasmid database, because this is the type of search experiments that was previously used in the mixing paper and was used to demonstrate something that was not previously possible, taking a good reference curated database of plasmids and comparing it with a comprehensive collection and identifying the plasmid presence-absence. In our case, we not only identify the presence-absence of plasmids, we can even obtain the full alignments. And the speed, we managed to go from the original approximately 2,000 hours, CPU hours, to approximately 74 hours. So this is the improvement once you start using phylogenetic compression. And again, it's not just presence-absence, it's presence-absence and plus alignments on top of that. And in terms of real time, it's when you run it like on your computer, it's searching for this specific data set is approximately 11 hours, which is quite reasonable, given that in how big database you are searching. If you want to compare it to something, compare it to how much time you need for searching in BLAST for like one query. That's like a good comparison because that's also big database, even though much smaller. And because sometimes people say, okay, so in the real-time search, we get the results almost immediately, but it's important to keep in mind the size of the database in which we are searching. So, and this is just like a proof of concept of another like possible application and something that what we would like to look a little bit more in the future. You might be able to align entire nanopore sequencing experiments to 61K. So we have example of one of them, 100,000 reads or 150,000 reads, only 4 hours real time on quite normal computer. 26 so total CPU hours across the cores. So this essentially means that mob search is fully suitable for maybe some future version. For instance, for point of care analysis, like in the future, you can really imagine that this is a picture from the Ebola project, from Ebola sequencing. And you can really imagine that in the future, you might be even like in a remote location, you might have a database of all bacteria ever sequenced. You do non-pore sequencing, and exactly on the same laptop, once you finish base coding, or maybe even during that, you might be able to search across all bacteria previously sequenced, even without an internet connection. So, some conclusions. So, phylogenies actually provide a universal organizing principle for genomic data, for compression and search. This principle can be applied, which we described, to compression of assemblies, various indexes, many different data structures. It's really very universal. It can also generate many different search protocols. The question is just how to design the right protocol, how to design the right batching, how to pick up and calibrate properly the low-level tools. There are many interesting questions around and open problems, and I'm listing just several of them, but there are much more. Of course, real-time search, we focus in our work mainly on like search with batches. The question now is if we say, okay, we won't work on laptops, we will work on some cluster or on some like high-performance computer. How to do real-time search. And it will lead you to completely different choices in terms of like batch sizes, low-level algorithms, and so on. Then how to search directly in the compressed domain. From the results that we have in the paper, especially in the supplementary, we know very well that the method in principle can be combined, for instance, with the Reaper algorithm. The difficulty of Reaper is that it's not currently very well scalable and it's not very well maintained. But in principle, we know that it can be combined with these algorithms that in principle allow random access. And they have very similar scaling properties as the exact algorithm which we are using in the core. So in principle, this should not be hard. There will be a lot of engineering effort. One fundamental question, how much more can we compress? And there are many relations to like string attractors, the delta measure, and so on. And our hypothesis is that not that much. I mean, we can get some gains. But for instance, if we go now from the size we are having to, let's say, 1/2, for instance, it's not a big change. It won't be like on the exponentials when we are like seeing the improvements of memories and so on. This will be just a shift by some constant amount of time. So, but still, it's a like very interesting question, like how much more can we compress this? Also, like much more general question is the proportion of the total microbial diversity that has already been sequenced, because this can tell us something about how much space we might need in the future. By the way, we can expect like huge change, like in the trade-offs, once big collections of metagenomic assembled genomes will become available, and then like the diversity will rapidly grow, and then we might assume that it will like get on more reasonable curve later. But we will see at some point like big change in the properties of data. Also, applications to metagenomics. We currently are working with isolates. In principle, we can think about ways how to make this work for complete metagenomes. So that's another big question. And also, like how to develop optimized tools. Like everything what we provide are just proof of concepts, just to show the feasibility. Like when you like the whole pipeline for searching, it's just Python. It's Python and gluing several tools together, and it's not optimized at all. If you like develop this in C++, it will work much faster. So thank you very much for your attention. I would like to thank all the people who participated in this project. It actually took several years, and we are very happy that now the preprint is online. So please reach out if you have any questions. Or any ideas, or we are still working on the materials, we are still improving the software, we are now working on a revision of the paper, so soon there will be a new version. And I'm very happy to answer your question. And I would like to especially thank Michael Bain, who's my PI at Harvard, and that I could work on this project, because it was very like high-risk project kind of. For a very long time, it was very unclear if there will be any results. And I got all the support from him, like various computers, and it was in the middle of pandemic. Then essentially nothing worked, and I must really say that I got from him the best possible support. So thank you very much. Thank you.
Thank you, Karel. Jens, I saw you raised your hand. No, you didn't. Who I have a question. I see many people raising their hands. So, Lynn, to start.
Yes, thank you. Thanks for the very nice lecture. Very interesting. But for this, for having these phylogenies to guide, say, or to as the organizing principle, you have to compute the phylogenies, right? Which is still a from a theoretical point of view, at least quadratic type of things to be just to know the distances. And second related question is, you speak several times of from left to right, but in phylogenetic, there is no really left to right, right?
Yes. And there are very interesting mathematical phenomena. I didn't speak about this. It would be like almost for like another lecture. So just to answer, like in reality, we use mesh tree. We did a lot of evaluation with two phylogenetic trees, inferred using state-of-the-art methods for bacterial genomics. We did with including like correction for recombination and many other things. It works slightly differently for assemblies and k-mers. For really when we are working just with k-mers, then the assemblies from mesh tree works slightly better. If we work with assemblies, the real phylogenies with like correction for recombination and so on are better. Of course, it does not get like exact, like let's say distances which we will be working with in computer science, but these distances are very highly correlated. And even though we might be able to improve a little bit, it gives us like most of the improvement comes already from this. Now we could think about like mathematical formalization of individual compression methods, studying it as an optimization task, and then looking how much goes to the optimum we get with this. And this is actually one of the ongoing projects, and we soon will have results about this. But for that, we need also, if we want to prove anything mathematically, then we have to fix the model of evolution and other things. But let's say that there are very profound mathematical phenomena behind. In phylogenies, yes, there is no left to right, and in principle, if you do any rotations, for instance, in the tree, it does not matter. Let's say that soon we will have a paper about this, where we will discuss this in more details.
Thank you, Lynn. Are there any other questions? Other questions? I saw some hands, but I don't remember whom it was. Or you can ask Brona?
Yes, hello, thank you. So I wanted to ask, do you have any ideas how this would scale to eukaryotes?
It's a good question. We have never tested it. Eukaryotes are subjected to very different, let's say, laws or maybe laws, but phenomena in terms of the population genomics. In general, for eukaryotes, I think we would need to use completely different types of approaches. Depends on the type of eukaryotes, if it's higher eukaryotes or lower eukaryotes. For higher eukaryotes, for instance, for the human genome, the diversity is actually very low, so it's a very different game. On the other hand, the genome size is big. So there are papers about that from Bell McKinnon, for instance, who needs to find ways how to kind of like split, for instance, by chromosomes and then compress the chromosomes like individually. For instance, yeast, it's a good question. We have not studied it yet, and it might be like one of the future directions.
And maybe I have one more question. So we were mentioning that in the data set, they use the same assembly for all data. And is it really important in your work? I understand that you need to filter on quality, but is it really important that the good quality ones are all produced in the same way?
In some sense, yes. For the search, it's extremely important, and we know it from other works on like rapid diagnostics of antibiotic resistance and so on. From the reason that very often the distances between the genomes, like epidemiological distances, can be extremely small. Sometimes there can be like one SNP difference. If you use inconsistent approach for assemblies, but I mean, up to some degree, it's inconsistent just by the fact that it's sequenced at a different place. But if you use inconsistent approaches for assemblies, for instance, like additional signal, and then you are unable to distinguish between very similar genomes. So that's the reason. Like in some sense, we are trying everywhere within the project to like limit the degree of freedom of failures, and this is like one of them.
Thank you.
We are getting a bit short of time. Maybe a last question. Sorry for this. I have one. If nobody has, just for me, there is something that is quite fundamental: is the notion of dynamicity. of the indexes. How can you add some new genomes to
Yes,
this structure?
very good question. Actually, it can be very simple or very hard. It depends on the specific formulation and your specific interest. In principle, our methods can be kind of like semi-dynamic. I didn't talk about all the details about the batching, but from some good reasons, when we split into batches from clusters, we sort it according to time. And there are good reasons for doing this exactly this way, because similar things are very often sequenced at the same time. So as a heuristic approach, if we preserve the accession order, it's a very good strategy for going from clusters to batches. And this actually means that since we have, for some species, like Salmonella, many batches that are kind of like closed, we can consider them already like fixed and never changing. And we can be, in theory, recalculating just the last batch of every cluster. Still, we need to recalculate that. We need to, the main issue is phylogenetic. Like, in theory, we might think about, if we were on the level of like, let's say, not like algorithms like our implementations, but like on the level of theory, we might think about like phylogenetic placement algorithms and so on. And we might study it even from like the theoretical viewpoint and what would be like the complexities and so on. From the practical perspective, the main difficulty is that if we have like a new genome which we want to insert, it goes typically in the middle. But at the same time, it would affect just the latest latest issue of each each species, latest batch. So in some sense, we could end up with something like what I would call like a semi-dynamic index, which is dynamic and is not at the same time. But at the same time, since it's like almost perfectly parallelizable, like everything, it's very easy to recompute the whole index from scratch. As long as you have sufficiently many nodes, it's trivial. Okay. It's like overnight, for instance.
Thank you.
Thank you very much for the invitation and also for the questions.
Is there a last question?
Okay.
Or not? So in this case, thanks again, Karel. Thank you. Thank you all. I don't know if you see me, but anyway. Thank you all, the participants, this nice audience, and see you soon. Goodbye.
Bye-bye.
Thank you.
Bye, thanks.
Bye.